name is Bianca Melita Palmas. I'm here to present my thesis project, Ovarian follicle count as a potential reproductive toxicity endpoint in small cetacean, an exploratory study of the ovarian forming function in harbour porpoises and common dolphin. This work was undertaken at the Marine and Freshwater Research Centre at Galway Mayo Institute of Technology and founded by the Irish Whale and Dolphin Group, the Regional Vet Lab and GMAT. Before going into more detail on my work, I would like to give you a little bit of background. So persistent organic pollutants are lipophilic industrial compounds, including PCB and the insecticide DDT. They are of global health concern due to their toxicity, high mobility, and resistant to degradation in the marine environment. Marine mammals are long-lived species and top predators. They are exposed to high level of this compound that accumulate in the lipophilic subdermal blubber due to bioaccumulation and biomagnification. Previous study on small cetacean highlight that the combination of POPs with other stressors are the cause of infectious disease, endocrine disruption, and reproductive failures, which are responsible for the decline in the reproductive health and the population status. However, studies on reproductive toxicity in small cetacean are still hindered by the lack of reproductive toxicity endpoint, resulting from a still poor understanding of what is the normal ovarian form and function of the species. Differences in ovarian performance between left and right ovary are common in mammals, including small cetacean, where the total number of ovarian corporal scars show an ovarian asymmetry in the ovaries of harbour porpoises, where only the left ovary appear to be functional. Conversely, common dolphins show an asymmetry of the ovary, in which both ovaries appear to be functional. However, we don't know yet whether harbour porpoises show asymmetry after birth or does it develop with the age. So to answer this another question, we apply ovarian follicle count technique also to provide important information about the function of the ovary of the species and their process of maturation through the ovulation. Starting from the ovarian reserve, which is the pool of primordial and primary follicles and other structures such as naked ova. And also ovarian follicle count technique is identified already as an endpoint of reproductive toxicity in humans and other mammals, where the high level of POPs negatively affect the number of follicles. It is applied on a set of neonate and juveniles individuals because in their immature ovary they show follicles in no stage of growth and atresia, and also because they possibly represent a linkage between reproduction and development. So by reviewing the appropriate ovarian follicle counting techniques, our objectives are to assess ovarian asymmetry between left and right ovary using follicular count, investigate on follicular growth in relation to individual growth by measuring the diameter of different follicular stages, and investigate the application of follicle count as a potential endpoint of reproductive toxicity in small cetaceans. Ovarian tissue sample from 35 sexually immature famous harbour porpoises and common dolphin were obtained from the Cetacean Stranding Investigation Program in UK and the Irish Vertebrate Necropsis Program between 1991 and 2019. Those two programs provide also data of the post-mortem analysis of each individual and included the cause of death category into infectious disease, trauma, and others, the body conditions, the beaching location, which allow us to display on a map where stranding were recorded, and the body measurement. So the reproductive tract of each individual was previously fixed in formalin, and the ovary, left and right ovary, were extracted, photographed, and measured, and ovary were dissected with a scalpel, and only the middle third of each ovary was selected for the histological preparation, which can briefly be summarized into the dehydration, clearing, and infiltration steps into the vacuum tissue processor the paraffin embedding, the microtome sectioning, the staining using the hematoxylin and eosin protocol, until the final microscope analysis of the final slide produced. More in specific, during the ovarian dissection, the middle third of the ovary of some samples was further dissected longitudinally, and each half was cut in 5 serial sections of 100 microns apart, following the latest toxicity protocol. The remaining samples were arrived from a previous studies already embedded in paraffin blocks. So just a random section of each middle third was cut into 7 microns. The imaging analysis followed and was performed using an optical microscope and cell size dimension software, which allowed to produce stitched images of the entire section of the tissue in the slide. So the images were saved and transferred to imaging software, and where an observer identified the different structures, so the different follicles classes, by using strict morphological criteria, and with the use of the multi-point tool on images, manually counted all the follicles showing a visible nucleus. Also, a subset of follicles was taken for the measurement of the 2 diameter, cross-sectional diameter, using a set scale and a straight line, always on MJ software. So we have 4 research hypotheses in this study. So is ovarian asymmetry in porpoises observed in follicle count from an early age? So we expect to see a higher number of follicles in the left ovary compared with the right ovary from an early age in porpoises. Is ovarian symmetry observed in follicle count in the common dolphin? Here we expect to don't see significant differences between the left and right ovary in common dolphin. And is there a decrease in total follicular count with increasing age? Here we expect to see that neonates show higher number of follicles compared with juveniles in both species. And to finish, is there an increase in follicular diameter in relation to body length? We expect to see different trend of follicle diameter depending on the follicle class in relation to body length. So on a sample of 35 individuals, we are collecting data, and we are testing normal distribution of values and homogeneity of variance in order to apply the best statistical model based on the raw data we have available. So far, however, in general, actually, we look at the follicle count within species to see differences in total follicle count between left and right ovary in the same age group and among age group. Here we applied nested ANOVA design to compare means for the data we have available. We also compared the section in the ovaries, so we look at the distribution of each follicle class in the ovary, and we applied nested ANOVA so far with the data we have available to compare the mean number of each follicle class in each section. I remember we have 5 sections in the ovary, the middle third actually. Also, we look at follicle growth and within species, and we are interested to see the follicular growth in relation with body length and other factors affecting the total follicle count within species, which are the correlation between biometric factor with total follicle count using pairs product moment correlation. And to finish, the association between total follicle number and independent variables such as left and right ovary, age group, and cause of death class. So our preliminary results show that there is a significant effect in the section on the presence of all classes of follicle between left and right ovary in 2 juvenile porpoises. So we can see in the graphs that the most central sections show the highest number of each class of follicle in the ovary. And also we see a significant difference in the total number of follicles between left and right ovary. I'm so sorry. We see, in fact, in this graph that there is also a decrease of the mean follicle count with increasing age. So we see juveniles with the smaller mean value of follicle count. And in fact, we see a negative correlation in the total number of follicles with increasing body length. In three porpoises, we can see that actually the harbour porpoises with the smallest body length of 70 centimeters show the highest number of follicles, while the other, which are juveniles with under 10 and under 20 respectively, show a decrease of follicles in both left and right ovary. So our data so far suggests that there is the total follicle number decreases as age group increases, according to previous studies, and the section in the ovary show a potential effect on the presence of the follicle classes. Those two results have to be evaluated in other individuals before making any assumption. And as well, naked ova are seen in both neonate and juvenile harbour porpoises, but have not been found in common dolphin yet. So it's an interesting result and has to be evaluated in the rest of the individual as well. And also asymmetry is observed in harbour porpoises ovaries from an early age by assessing it on the smallest individual in our data in our sample sites, and appear to persist with increasing age group and body length. So far, our results, our findings are in line with our hypothesis. So some strength and limitation of this technique should be highlighted, such as that great coverage of the ovarian cortex and minimized overcounting of follicles. So we are interested to see difference in the result with this and the other technique available in the studies coming from a previous study. And this technique follow a strict protocol in which reproducibility and high level of agreement between observed can be achieved, and we can calculate Kendall coefficient of concordance. And however, it's possible to come across highly decomposed and damaged tissue which produce outliers, so it's recommended to use freshly dead and unfrozen samples where possible. And also the histological technique is quite costly and time consuming, and this needs to be taken into account where planning a project and the budgeting. Overall, ovarian follicle technique, counting technique, has the potential to be a good technique to assess ovarian form and function, and has the potential to be a good endpoint for reproductive toxicity. So to conclude, I would like to share with you some troubleshooting and general recommendation from my personal experience with the histological technique, especially when staining. Avoid uneven hematoxylin and eosin staining, and ensure complete nuclear bluing in order to identify the follicles. Renew reagents regularly, so in order to avoid uneven results and maybe have to throw away important samples. Discard the section with folds. And because histological technique follow many steps, it's sometimes difficult to identify the origin of the issues. So my recommendation are to take note of any change in the processing and go back through each step and repeat it, but don't use good sample for these steps. And practice with those samples so you can make your own technique. Make use of online resources such as panel discussion, manuals, and also YouTube video can be really handy. And then you will be able to produce a high quality histological slide. I would like to thank my supervisor, Dr. Sinead Murphy, and the really kind staff of the Marine Freshwater Research Centre and the Irish Whale Dolphin Group that funded this project, and the MBRC family as well. If you have any questions, please feel free to contact me, and thank you.